All right, my name is Takuto. I'm one of the developers for the GATK, and we're going to talk about the next step in the workflow, which is genotyping, genotype refinement. So this is where we are in the pipeline. We just filtered the variants using the CNN and or VQSR, and now we're going to get a better calibrated probabilities for each genotype. So why do we care about genotypes? And before we get to that, so what I mean by genotype is, let's say, at a particular site, the reference is a C, and then so it is a genotype is the allele, the alternate allele. Is it a SNP to C to T, or is it C to A? So there's the allele part, and there's also the number, the count of alleles. By that I mean, is it a homozygous reference site, or is it a heterozygous site? So one copy is the reference, one copy is the non-reference. Or is it the homozygous variant, the two copies of an allele that does not match the reference? So that's what I mean by genotype. And that's, although I'm not a clinician, it is important to get those right. There's a huge difference between there being a not variant, so homozygous reference, and there being a variant. But also, it is important to distinguish between whether a genotype is a heterozygous or homozygous variant, whether they're one copy of each allele or two copies of the variant allele. Perhaps the number of variant allele encodes or shows up in the degree to which a patient is affected by the disease, or it can also affect whether the child of the patient can inherit those alleles. So it's important to get the genotypes right. And it's also the counting the alleles is important for doing population genetics. So here is an IGV screenshot where you can see the genotype being encoded in the VCF file. And here, we're looking at a site where the reference, the human reference is C, and we're looking at a C to T SNP. And the workflow found this variant, and it says the genotype is two copies of the T allele. So this is a homozygous variant site. And it does say homozygous variant for home var. Okay. And so the genotype call quality, the genotype probability, it could be the data that we got, could be perhaps we got bad data at a particular site where it didn't get enough coverage or something for a multitude of reasons. The confidence with which we call a variant could be low. The probability of there being a particular genotype could be low, or perhaps sometimes we can't tell the difference whether the variant is a heterozygous or homozygous variant, for example. And the idea of genotype refinement here is to leverage some independent resources, such as 1,000 genomes or more likely NOMAD or GATK, to improve upon the confidence with which we find these variants. And also, we can, if it's available, we could use the genotypes of the patient's or the sample's parents to see if there's any what's called the Mendelian violations, where the parents' genotypes do not agree with the child's genotype. I'll give a specific example later. So we could leverage that information too. And using those pieces of evidence, we can update the probability of each genotype to get the posterior probability of genotypes. And that, using our prior knowledge from orthogonal databases to update the probabilities it sits on the statistical machinery of the Bayes rule, and this is just the equation that encapsulates Bayes rule, and the idea is to update your belief, update your prior belief based on what you observe, the data you observe. So for instance, if somebody, let's say you work at an office with no windows, and you slept under the desk the night before, you wake up, and your coworker shows up with an umbrella. Now that you've seen that a coworker walking with an umbrella, what's the probability that it's raining outside? Well, the inference really depends on like where you are. So if you live in Boston, right, maybe it rains like once a week or something, less than that, once every 10 days. So it's like a moderate prior probability of rain. But if you lived in LA, for instance, right? Although I've actually never been to LA, but I've heard that it doesn't really rain much there. So it's the prior probability of rain in LA is significantly lower than in Boston, and perhaps the coworker just came from an audition for a part of Dancing in the Rain or something, right? So that, so then there's the posterior probability of rain would be different depending on the prior. So likewise, if you have an allele that you rarely ever see in the population ever, like if you have a mutation at that particular locus, perhaps you mess up with ribosome or something. Then you never see those. You never see a mutation there, but then you happen to have a sample where you see this mutation. So it's likely it could be real, or more likely it is just some sort of sequencing artifact or something. So you can leverage the information from the population orthogonal data set to update your genotype quality. So here's the workflow. We start with the recalibrated filtered variants, and we use the population allele frequency priors, and if you have it, the family trio information to get the posterior genotype probabilities. And then once you have that, you can do some further filtration based on the posterior. So now that the genotype probabilities are updated, perhaps we could filter, get rid of the ones that get low probability, the genotypes that are whose probabilities are low. And now you're left with the high quality variants. And you could also optionally annotate if the family information is available, then we can tag the variants that are de novo. De novo variants are variants that only the child has, but neither of the parents do. So this is a workflow. And this is how you run it on GATK. Calculate genotype posteriors is the name of the tool. The pedigree file -pd is optional. Are there any questions so far?
What is the format of such a pedigree file?
It's a .ped file. If you just Google it, there's a GATK post. Yeah, it's like a table, like a text table of, yeah. Okay. So here is a example of a genotype. So here we have a data with some evidence for alternate allele, but not all of the reads are the alternate allele. So alternate allele here is the red. Gray means matches reference. Okay, so the baseline. So without using all the steps before this refinement step, the workflow called HOMVAR, homozygous variant, meaning 2 copies of the alternate allele, which is reasonable. They're over maybe like 70% of the reads are the alternate allele. But then if you look at the allele frequency, so if you look at across the populations, this allele seems to be not that common. So it's unlikely that although it's possible, it's unlikely that this particular sample had 2 copies of this rare allele, and plus like the genotype quality, so the probability of this particular variant being homozygous variant is not that high. So you can see that here. So this is a log. So these are the probabilities of these are the relative probabilities of each genotype. So 0 and then taken normal. These are relative probabilities where the genotype with the highest probability is 0, and the other 2 is like the negative log 10 of the other probabilities. So in this case, the HOMVAR, the head call was 10 to the negative 3, a factor of 10 to the negative 3 less likely than the HOMVAR call. But then after applying this prior information, so 0 is again the highest genotype with the highest probability. So now we updated our beliefs and concluded that the het is a more likely call, and the next most likely call, which is homozygous variant, is not very likely. Okay. And this particular sample that we're looking at happens to have a truth data set, and it actually agrees with the truth. So that's like the correct call. Okay. Another example. This is the opposite of the example that we just looked at. So the workflow up to here called that this variant is a heterozygous variant, but with not much confidence, because it's actually saying that the probability of heterozygous and homozygous variant are they're equally likely. So it doesn't know which one's true. But then if you look at the population, it turns out that the frequency. So almost everyone has 2 copies of this allele. Turns out. So using that information, then it's more likely that this person too has 2 copies of this variant allele. Yes.
What does the Q stand for? Is this the Fred score?
Yeah.
Isn't the higher Fred score usually meaning that you have more
Confidence.
Base accuracy, so that you're more confident?
Yeah. So it improves. So Q0 means like it's a toss up. It's a and Q0 means that if you take the ratio of the most likely probability to the next most likely probability, it's 1. Okay. And Q16 means if you take the ratio of the most likely to the next most likely, you get 10 to the 16th power. So it's, yeah. No, 10 to the 1.6 power, sorry.
So here, the heterozygote one in the posterior probability is the most probable one. We will take the heterozygote in this example.
In this slide, no, the most likely one is the homozygous variant.
But isn't the heterozygous one the one that has a Q16?
No, so yeah, sorry. So it's not quite sure. So this is, I can actually talk to you offline and describe to you how this works. But this is pretty much saying if it's a relative probabilities of each genotype. So you take the probabilities that add up to 1, divide everything by the highest probability. Okay, so you get 1 and something very small and something very small, and you take the negative log 10 of each number. So 0, so log 10 of 1 is 0. So yeah, it's a nitty-gritty thing that we can talk offline if you're interested. Okay. So yeah, so we can see the kind of effect running calculated genotype posteriors have on the quality of the variants. So here we're looking at, on the x-axis, the reported genotype quality, which is what we what the workflow calculated, and blue is after applying calculated genotype posterior, and the red balls are the one before applying this calculated genotype posteriors. And on the y-axis, we have the empirical genotype quality, which we can calculate by using the truth set and looking at all of the variants that had genotype quality of 10. So genotype quality of 10 is that you make a mistake, 1 in 10 variants will be an error. And you can actually look at all the variants that we gave a quality of 10 and count how many of them are actually wrong if you have the truth data. And that's what's plotted here. And as you can see, so if what you reported, what the workflow reported, and what it actually is matched, then they should be on the diagonal. So as you can see here, that the blue balls, the posterior probabilities line up pretty well with the diagonal, which is good. But before applying this, the homozygous reference calls had higher empirical quality than the reported quality. So the reported qualities were supposed to be higher, but they weren't. But then after applying this step, all the balls shift to the right, and not all the balls, but most of them shift to the right and kind of go along with the diagonal. And similar story, but in the other direction for homozygous variants. And here we're looking at the same data, but in a different view. Here we're plotting on the x-axis the qualities, so the qualities output by the workflow prior to this step, and then this step being the calculate posterior genotype posteriors, and this on the y-axis is the qualities we get after. And what we see is that, and the red ones are the incorrect genotypes, and the blue ones are the correct genotypes. And as you can see, the red ones stay pretty much the same before and after applying this calculated genotype posteriors. But then the ones that are correct genotypes, we have more confidence about the correct genotypes. So it's doing no harm, and giving more confidence to the already bad calls. But we're boosting the probabilities of the genotypes that were actually correct, but we weren't confident by a quality score of 10. So it's like a factor, by a factor, probability, improve the probability by a factor of 10. All right. And so that was the using the population allele frequency as sort of the independent orthogonal piece of evidence, prior evidence. And here, we can also use, as I said, the family information, the genotypes of the sample's parents. For instance, if your mother and your father are both homozygous reference, if the child has, let's say, is a heterozygous for this variant, so that alternate allele didn't come from his or her parents, so that it must have been what's called a de novo mutation, and that's very unlikely. So we can sort of leverage that information to improve the genotype qualities too. And here's some equations, but what to take away from this is that there's this factor mu, which is the probability that we get a de novo mutation. This is a tunable parameter, and the recent literature says that the rate of mutation in the human genome is de novo mutation in the human genome is about like 10 to the negative 8 or something. So we expect about 30 de novos. Is that true? 10 to the negative 8 power? Yeah. So we expect about 30 de novo mutations. And then like so MV here stands for Mendelian violations, which is a genotype that's inconsistent with the parents' genotypes. And if you have two Mendelian violations, for example, if your parents are both homozygous reference, but the child is a homozygous variant, for example, that's like two, like lightning struck twice. So that's even less likely that happens. So if you multiply 10 to the negative 8, if you raise 10 to the negative 8 to the power of 2, you get 10 to the negative 16, which is very unlikely. So we use this information as priors and update the posterior probability of a genotype. And once we applied both the family prior and the population prior, we get the updated posterior probabilities of each genotype. And now that we have this new and updated genotypes, we might want to filter some of them that turned out to be low quality, and this is how you do it. The GATK tool is called the variant filtration. And here you can see you can set this 20 to whatever parameter you want and filter any sites, any variants with quality lower than Q20, for instance. So that way the data that goes into further analysis further down the pipeline will consist only of higher quality data. And we also give an option if you want to tag each variant that is a de novo variant, since those are rare and could potentially be interesting to look at. You can annotate those variants with the variant annotator tool. And a show of hands, like how many of you are familiar with the de novo mutations? Okay, one or two. So de novo, it's just de novo mutation is a mutation where parents, the blue here means, or blue is one allele, so both parents have 2 copies of the blue allele, but the child somehow has this orange allele that he or she definitely did not inherit from his parents. So it must have happened. It must have happened sometime after somewhere in the biological process of this person being a human, and that's rare. We only expect about 30 de novo mutations.
How do you estimate this mutation rate?
How do you estimate the 10 to the negative 8? Yeah, I think the one I looked at was there was like a study about there's a study in Iceland that looked at a lot of different trios, a lot of parent-child trios, and that's what these, and they counted the number of mutations that were only in the child, and then that's the rate that they came up with, I think. Yeah, and de novo mutations are, novel, rare, and yeah, so it could be if the parents, neither parents has the disease, but the child has, could be de novo. I'm not a clinician, so I don't know, but it seems plausible. And here is a real case study in which we started out with, in the sample, we started out with about 400 de novo mutations. So 400 mutations that only existed in the child, but again, we expect about 30, so this seems really high. But then after we applied the prior information about the populations and the pedigree, so that number went down to 17. So we got rid of a lot of probably spurious false positives by applying this calculated genotype posteriors. And after even filtering even further, we ended up with 8 de novos. Okay.
Is GQ just quality filtering? What is GQ?
GQ is the genotype quality. So earlier we set it to 20, which means that it's the same thing. It's saying that if you're
Oh, so it's a parameter of the variant filtration.
it's a parameter of the variant filtration that you can when you type this command up, you can change it to anything. Yeah. Yes.
Is it an average of the genotype qualities of individuals on that variant? So you have the genotype quality as a field in the for every person, right? In the VCF file, we'll have like a depth of coverage, genotype, and genotype quality. So is this GQ an average of those as a metric of the variant?
I see.
Or are we filtering every individual genotype based on the genotype quality? Or how does that transfer to the variant?
Yeah, so that's a great question. So the question was, so each sample in the VCF has, so for each row in the VCF file, each variant has many samples, and the genotype quality is, each sample has genotype quality. One sample could have 30, and the other sample could have something lower than 20. Then how do you filter? Yeah, there must be some sort of, I don't know. I can look into it, but.
So basically this metric that we're setting up, GQ less than 20, does this apply to every sample, or does this apply to an average of those estimates for this variant? So
No, it is per sample. So when you apply that filter, it's going to annotate for that particular sample in its VCF fields. It's going to annotate a filter that says this particular genotype for this sample is considered low quality.
if my VCF is multi-sample, so like it has multiple samples, will it annotate every
Yes.
genotype with the?
Yes. And the processing is done, I forget what the logic is for, in terms of how the processing is done logistically, but that the algorithm is applied per sample of interest. Yeah. So one sample's genotypes are not going to influence an unrelated sample's genotypes. Does that make sense?
So these VCF files are individual VCF files, not like mother, father together.
Let me grab the mic. Sorry. Yeah, so so each sample has a GQ value, right? When you run the refinement steps, the refinement step acts on each individual sample. And when you apply the filtering, that the annotation is applied in a new genotype filter field for that particular person. Now, to be honest, I forget if the genotype refinement tool, allow you to put in a multi-sample VCF. I believe it is the case. So what will happen is that effectively you have that annotation that's added per sample. Does that make sense?
So I think
Is that helping?
there's a different. So you're thinking about filtering as like filtering a variant, like a row,
Yeah.
like in the filter column of the variant.
So I'm trying to understand, are we filtering out the rows or are we?
No.
I think we're, yeah, it seems like
The
we're
columns, yeah, columns.
we're filtering. We can take this
Yeah.
offline, but it seems like, yeah, is there like a tag or something for each
Yeah,
like a variant
exactly.
field, genotype field?
You have the column for that particular sample, and it has the DP for that sample, and it has the GQ for that sample, and all that stuff. You're going to add, I think it's FT for filter, and that particular thing will be specific to that sample. So we're no longer looking at the full row. It's really per column. So per cell in your spreadsheet, really, if you think about it that way.
Yeah.
Does that make sense?
Yeah.
Cool.
Great. All right. Any other questions? Where were we? We were here. And yeah, so the mu value, the 10 to the negative 8 number is just a suggested default. But if you happen to have a call set where you expect there to be a lot of de novo mutations, for instance, then you can bump up this number. You expect there to be, higher number of de novos, then you can bump up this number and then increase sensitivity, meaning that you can discover many more true de novo mutations at the expense of increasing the number of false positive de novos as well, the false non-variants. But that's a trade-off that you have the option to make. Yeah, so in summary, genotype, we can leverage the family pedigree information, the family genotype information, as well as the allele frequencies in the population to update, and using Bayes' rule to update the genotype probabilities. And based on those updated probabilities, you can filter the variants further. So that's all I have, and if you have any questions, I'll be happy to take them.